Uh, Namaste, everybody. Welcome to the third day of the Applied Bioinformatics in Agriculture and Medicine seminar. I'm really excited that all of you are able to make it today. I already see more than 120 participants for this demo session going on from today. So today we'll get into a lot more technical stuff, and we'll try to orient you all towards how we can use bioinformatics in our daily lives to analyze data. And in order to start the session for today, I'd like to invite Dr. Ananta Acharya to lead the program. Dr. Acharya.
Thank you, Dr. Paudel. Today it will be more interactive than last 2 days. So if you have any questions, please write your questions in between, and we'll try to answer as we go. And although I may be speaking in the beginning, and or Dr. Paudel or Dr. Parajuli or Mr. Subedi, we'll be interacting with each other, and we'll be answering whatever questions we see, and we'll try to answer all the questions you have. What we are also really interested is this is co-hosted by AFU Biotechnology Center of Biotechnology, and NAPA. What we are also, and I think we learned that, and when we look at our registrants, we see a lot of people from NARC or KU or other universities. So if you are not from AFU, can you please write in the chat box that you represent some other organizations, and so that we can also get some perspective of your organization, and we can also think about how can we collaborate over there. Okay, so with that, I'll start my slides. So this one is, we are presenting this in such a way that we are very big. Let's say we want to start a bioinformatics lab. We want to start a bioinformatics research in some biotechnology lab or some other lab that we have never been, that we are not really used to. So how do we get started with the bioinformatics research? And what we are focusing here, and you have heard from us yesterday and the day before, that in the bioinformatics there are 2 or 3 key components: doing core computational stuffs, databases, designing algorithm, writing softwares and programs, and the other one is applying all those programs for agriculture or medicine, microbiology, evolution, animal breeding, plant breeding, and those different stuffs. So our focus today is on the applied bioinformatics. So what we will focus on is, we'll use the publicly available softwares and algorithms rather than creating our own. However, we still need to know some programming knowledge. We need to have some database knowledge. We need to have some statistical knowledge to navigate around the data and report those scientific reports, publish, and get some insights from the data. We still need to know the programming and the statistics. So that is something I wanted to discuss here. So on the computational side, what we need is, we need some server, where most of the time the server, the operating system will be Linux. It should have a lot of enough computation power. We should also have some storage system. It has to be attached with the server, but it doesn't have to physically be in the server. And we have to have a lot of different programs. We also need to have a client computer. So to do basic bioinformatics, a client computer and a server computer can be same, to do very basic experiments, or something like Dr. Paudel was showing yesterday with Tassel. You can do in your laptop. If you have enough processing power, you can do that in laptop. So sometimes it can be same, but most of the time, to do whole genome analysis, we have to have a client computer and the server computer differently. Then in the client computer, we need to have a way to connect to the server. The way we connect to the server, if it is a Mac OS or Linux, we can directly use terminal. If it is Windows, we have to use some programs like MobileStorm or Putty or those other file system or the other different programs or Sigwin and those kind of things. And the third very important thing in the bioinformatics is visualization. How do we visualize the data? Sometimes the visualization can be done in the client computer, in your browser, or it can also be done with the server. So I see a lot of text messages or chat box popping up there. I'm not sure what they are because I'm going through these slides. So Dr. Paudel or Parajuli or Mr. Subedi, if you see something that you want to add in between while I'm talking, any questions or any of my talks, please feel free to interrupt me and give your ideas here. Okay.
Yes, sure. There are just a lot of participants from outside, so like out of
Okay.
AFU.
Okay, that's really great. That's really great. So now on the data, so we need the computational power. That's how we use we need computer, but just the computer is not enough. We need data. So where do we get that data? We can have our own lab, own sequencing lab. From the wet lab, we'll make the sequencing. We can have the sequencing lab. We can also outsource the sequencing. We can collect the leaf samples, different samples by ourselves, and we can send it to some outside vendors. Or we can use a public data, like what we showed you yesterday about the coronavirus or the rise. Those are the data. They are not created by us. We just went there, found those data, and then try to get some knowledge from that one. And that is what I talked on the very first day, that we don't have to start with some hypothesis. What we can do is we can start from data and generate our hypothesis. So for example, Dr. Paudel yesterday looked into the around 44, 45 rice varieties. We didn't have any hypothesis, but when we looked into the data, then we started, oh, okay, we still see these 3-4 clusters. There are different basmati or aromatic rice, but they still cluster differently. And that becomes, okay, that becomes the information. So that's how we can do. And the similar thing with the coronavirus example, right? That D614G mutation, okay, that mutation seems that was not even in Nepal very in the beginning, but if you look at a lot of data in Bangladesh or India now, you start seeing that mutation. So those kind of knowledge we can just gather from public data. So I think in the beginning, we can start, we can actually have some very good papers without actually generating any data. Now talking back about the server. So the server again can have two different ways. We can have on site or on premise. We can have our computational power over there, or we can actually rent from the cloud computing. For example, AWS or Azure or Google Compute. I think even Alibaba or other companies also have this cloud computing platforms. So in our session, we'll be using Google Compute session. And then it needs to have a lot of different programs. And Dr. Paudel, can you please look at the annotation and clear the annotations, please? Seems like somebody's annotating. And then we have the requirements, and in the requirements, we need to have a Linux operating system or some mixed flavor. So even Mac will work. We need to have some programs, which I'll come back later about different programs, basic programs versus bioinformatics software. Then the big challenge is the storage. Think about Dr. Bartola's talk 2 days ago, and she was presenting the genome size. And if you remember the genome size of coronavirus from yesterday, from COVID-19, it was about 30,000 kilobases. So very in the bottom left here, that was the size of the coronavirus. Very here, just this is already 10,000. So this is 10,000 kb. So this is in kilobases. So this is already in 10,000 kilobases. So that is even smaller than what it is shown here. Then you go to bacteria, it is a little bigger, fungi, algae, and so on and so on. Mammals, it is saying about 10 to the power 6, 2 to 3. So human are about 3 billion, right? So that's where most of the mammals are. And look at the flowering plants. It has a huge range. And if you look at the wheat or oat, they have huge genome. Think about that 3000 rice genome just with maybe 5 to 20x coverage. Even when the data was compressed, the size of that data was just for the raw data. Forget about the mapping and SNPs and BCF and those stuffs. That was 15 terabyte. So storage is a huge problem in bioinformatics, and especially intermediate files. If we are not good about cleaning intermediate files, we will fill up those spaces very quickly. So again, on the storage, we can have a on-site storage or cloud storage, and this can be very costly. The good thing about the compute engine is you can rent and you pay as you use it. So you can rent a cloud, and then you are not using it, then you are paying very basic minimum charge over there. But for the storage, you are always storing there. So that is why sometimes the storage cost is actually higher than your computing cost. Then network, really important. We need if the server and client computers are not same, then we need the client server connection. We need to download and install a lot of different programs. We need to download or access different data wherever they are stored. We need to upload the data and visualize the data. So a network, a very stable network is also another really important thing. Then on the softwares, on the software side, on the server, it should have general, and the general, I don't even talk about the OS and what is included in the OS, so that would be included. It should have some programming languages, and any new beginner programmers right there, I'm a fan of Python, and I really suggest you start using Python if you are never in the programming language. But if you have knowledge in other programming language, you can use any of those. Paul was really used a lot in bioinformatics, so that is why you still see a lot of legacy programs in Paul. So that is why you need Paul. And R is a statistical program. It's not a programming language itself, but it is also a programming language, but it is mostly used for statistics because of the packages. In Python, you also have the packages, but the statistical programs and the visualization packages you have in R, that is unparalleled. Then you can also have Conda or Docker environment for reproducibility of your different programs, different versions of program. One of the problem in the Linux is, I know even in Windows, sometimes you get this program is ready to update, you want to update it, you will just click yes, and then it will update most of the time, right? In Linux, it will almost never ask. And then you are working in one version of one program, and 3 years later, you are still working on that version. But another program wants a result from a newer version that doesn't work, and those kind of things. So for reproducibility in Linux, we really need to know what version of the program we used. And then there's the version control on the script. So you write some script, and a lot of the bioinformatics is hit and try and error. So you write some code, you have different parameters. So let's say you are mapping some sequence with the reference genome. You want to say, oh, okay, this is not very close organisms, so I want to allow many mismatches. But after you see the result, this doesn't look good. Now I want to be very conservative, and I wouldn't allow a lot of the mismatches. Then that case, you will write a different code. And if you have that version controlling of your codes, it will help you what you did in earlier versions. And a lot of bioinformatics software, we cannot list all the bioinformatics software here. I do not know all the bioinformatics software, and I doubt there is anybody who knows all the bioinformatics software. And one of the example is probably all 4 of us here probably have written more than 10 or 15 bioinformatics software by ourselves. So there is almost impossible to know all those different things. But there are some really popular ones, and there are some really basic ones almost everybody uses. So downloading the sequence from NCBI, you can just click and download, or in the Linux, you can use the Acharya tools. There is some QC software. There are some adapter trimming softwares. There are some alignment softwares. You have heard a lot about probably a BLAST or some pairwise alignment with MSA or Muscle or Cluster. So those kind of programs. We have a mapping software called BWA or Bowtie or Minimap2, so which will align our sequencing reads to the reference genome. Whenever those sequencing reads are mapped, they would be called BAM or SAM. So we have these SAM tools and BAM tools. we have heard about the variants like indel, snip. So for those purpose, we see the GATK, BCF tools, SAM tools kind of programs. For visualization, we have IGB, JBrowse kind of programs. And similarly, all the other tools like the bed tools, BCF tools, Blink, Top Hat, or Coplinks, and many more. And so we'll use some of these programs tomorrow and next day, going through some of the program here. On the client side, this is something I wrote in the chat box earlier, MobiXterm. That is something we will be using to connect to the server. So we already have the Google Compute server. So if you are familiar and if you want to use it, start downloading the MobiXterm, and we'll let you know how to connect to that. Then for the file transfer, you can use the same MobiXterm, or you can have, if you are a Windows computer, you can use WinCP. And for writing the code, you can write in BIM itself within the Linux, or you can use a Notepad++ or Visual Studio Code, or there are even just the Notepad. Just don't use Word. Okay. And all, most, above all, everybody else, you need a bioinformatician, right? It's you. So who are you? You are either a biologist with the knowledge of computer science and the statistics and mathematics, or you are a computer program science student and a person in computer science with the biological knowledge and stats knowledge, or you are in the stats with some knowledge in the computer science or the biology side. So you have to have a really good understanding of one domain, but you need to understand what other domains are doing. And with that, I'll finish this talk. So we'll entertain any questions here, and we'll also, like, if Dr. Paudel, Dr. Parajuli, and Mr. Subedi, if you want to add anything on these ones, please join me.
Sure. If anybody have any question, you are free to unmute yourself and ask the questions.
It's really interesting to see a lot of people not from AFU or IES. That's really great.
Our reach is far and wide, so maybe we should move ahead to the next session.
Okay, then let's move to basics of the molecular biology, because unless we understand the molecular biology, we cannot do any of the bioinformatics. So I'll invite Dr. Saroj Parajuli to talk about basics of the molecular biology.
Can you see my screen?
Yes.
Okay. Welcome back, everyone. Like, most of you already know the basics of molecular biology, and I'm just going to refresh you about some of the important things that we need to learn for the sequencing purpose. And so this is one of my favorite slides, and this is central dogma of molecular biology. So if you understand this slide, then we understand most of the process of the molecular biology. So here is the flow of information from DNA to RNA and RNA to protein. So DNA is double-stranded, and the flow of information from DNA to RNA is called transcription, which is done by RNA polymerase. And information can flow back from RNA to DNA, and that is done by reverse transcriptase enzyme, and the process is called reverse transcription. And the flow of information from RNA to protein, that is called translation or the synthesis of protein. So if we see the DNA sequence, it has 5' to 3' in on the top, and it has 3' to 5' in on the bottom. So this is the double-stranded DNA, and here are exactly 30 nucleotides. That means we have 10 codons. So I have shown the codon in RNA. So AUG, CAC, AUU, all are the codons denoted by 3 letters, and these are the amino acid that is coded by those codons. So if you see the information from DNA to RNA, it is flowing in the DNA to RNA by transcription. And if we see the RNA to protein, then there are 10 codon. But if we see the final codon is the UAG, which is the stop codon, and the first codon is always AUG, that codes for methionine. And if we see the protein sequence, then it is the primary sequence. So there will be secondary and tertiary sequence of the protein too. So due to the stop codon, we have only 9 amino acid. 10 codon can code for 10 amino acid, but final codon is coding for stop codon. So we have 9 amino acid in the protein. And I have a question for all of you. I'm keeping the question in all the slides, and we can discuss at the end of the session. Can the information flow back from protein to RNA? So just think about this one. And let's talk about the structure of, I don't know, I cannot see the top of my slide. Just a minute. Okay. So let's talk about the DNA structure. So it consists of sugar, phosphate, and base. And DNA consists of different type of sugar, and RNA consists of different type of sugar. That is how they are named. DNA consists of deoxyribose sugar. Deoxy means the devoid of one oxygen. So if we see the deoxyribose sugar and ribose sugar, then the deoxyribose sugar that is in DNA has one oxygen less. It has hydrogen atom in the second carbon position, while the ribose sugar has hydroxyl group in the second carbon position. And it also has the phosphate bond, and the phosphate and the deoxyribose sugar, it makes the backbone of the DNA. So the question is, what is the charge of DNA? And out of the 3 components, we have covered sugar and phosphate. And let's talk about the nucleic acid, that is the base. So there are 2 kinds of base. One is purine and other is pyrimidine. Adenine and guanine are purine, while cytosine, thymine, and uracil are pyrimidine. And thymine is present only in DNA, but in RNA, the thymine is converted into uracil. When the transcription happens, the thymine is converted into uracil. So if there are any questions, you can type in the chat box, or maybe we can discuss when we go through the slide also. And let's talk about the base pairing in the DNA structure. So there are 2 kinds of base pairing. One is the AT, adenosine pairs with thymine, and guanine pairs with cytosine. So in the base pairing of adenosine and thymine, there are 2 hydrogen bonds, while the guanine and cytosine, it has 3 hydrogen bonds. And as you can see in the figure, this is the DNA, and the blue color represents the backbone, that is the phosphate and the sugar, while the colorful G, C, AT, these are the base. And let's talk about the structure of the RNA and conversion into cDNA. And the question is, why RNA is unstable compared with DNA? So I have taken the same sequence from the previous slide. So RNA, the flow of information is from RNA to cDNA. And this is done by reverse transcriptase enzyme. And when RNA makes cDNA, first the single strand is synthesized by using the reverse transcriptase enzyme. Then the second strand will be synthesized by using the DNA polymerase enzyme. And if we go to sequencing, I don't know if Dr. Acharya want to add something in the slide. Okay. So when we do RNA sequencing, first step is to convert the RNA into cDNA. Then the DNA will be sequenced. The RNA is not directly sequenced, but the RNA which is converted into cDNA is sequenced. And we have 4 different bases, and it forms the triplet, means like 3 base makes a codon. So if we do the math, then the 4 base and the 3 triplet code will make total of 64 codons. So in this codon wheel, you can see there are 64 codons, and 64 codons are coding for 20 different amino acids. And the start codon is always going to be AUG, and that is in the RNA, and we represent as ATG in the DNA. That codes for methionine, while there are 3 stop codon. So 3 stop codon are UGA, that is 1 stop codon, and second one is UAG, and the 3rd stop codon is UAA. But if you see the sequence of DNA, then you have to convert U into T. So it will be TAG, TGA, and TAA. So so what is gene, and how big a gene can be, and what structure are needed to make a gene? So I'll discuss these questions in the upcoming slide. So this is the structure of a gene. It consists of a promoter sequence, then it has 5' UTR, then the start codon, exon. It can have multiple exons or multiple intron. Then finally on the final exon, it has stop codon, and it has 3' UTR, and it has poly A tail. So when this is in the DNA sequence, that is for the gene, and when it is transcribed, it will have the 5' UTR and the start codon, exon, and the stop codon, and it will have 3' UTR. That is called mRNA or transcript. And if you remove the 5' UTR and the 3' UTR, then it becomes the CDS. CDS means coding DNA sequence. So only this sequence are going to be the protein sequence. That is from the start codon to the stop codon. So if you see the DNA sequence, the start codon and up to the stop codon, and excluding the intron, is going to be translated into the protein. And let's talk about the sequence of a promoter. And the promoter can be of any length, and on average it is around 1000 base pair upstream of the start codon, and it has Tata box, it has cat box, and it has some enhancer region. And when the transcription happens, the RNA polymerase is going to attach in the promoter region. And a lot of other protein like repressor protein or activator protein that can attach in the enhancer region, and that can affect the rate of transcription. So what a gene looks like. So our gene has a UTR, then it has exon, intron, and the terminator. So if you see the figure on the left, then this is the complete sequence of a genomic DNA. It has 5' UTR denoted by green. Then it has some intron, that is the white, and it has exon, that is, if you see here, this is the first exon, and this is the start codon, ATAC. This is going to be the start codon. Then after the first exon, it has the first intron, then it has second intron, and it has third intron, fourth intron, it has 5 and 6 intron. And the light blue color and the dark blue color, they are representing the exon. And finally, on the red color, we have the 3' UTR. So this is the genomic DNA sequence. But if you see the transcript sequence, then all the intron are splashed out, and the remaining portion are going to be the transcript sequence. And if you see the CDS sequence, that is translated into protein. So it only has the exon sequences from ATG to TAG. So this is going to be translated into the protein. So this is the final protein sequence from this gene. And this is a TF3A gene that I have taken from rice. So I'll show you how we can get this sequence from NCBI and also from phytosome. So I'll move to NCBI website.
While Dr. Parajuli is looking at that, I saw a question from T.N. Gosal. What about the cistron in eukaryotes? I think cistron is, as far as I know, is a kind of old term. Before we understood a lot of this molecular biology. It was basically the unit that transcribe unit from starting from the start codon to the stop codon, and so is used to be called cistron, but right now cistron is defined as gene generally, from that start codon to the end of the codon without any introns or the 5 UTR and those kind of things. It's kind of that coding sequence, CDS.
So if you want to follow with me, you can go to NCBI website and this phytosome website. If not, like we can show it later too. So I'll be looking for the same gene that I have shown the sequence, that is TF3A. So if I only type the TF3A gene, then I can see there are 406,067 different genes. Then I just click here. Okay. But maybe we just need a gene for a specific organism. So let's go to advanced and we can set our organism here.
Dr. Parajuli?
3A.
You are not sharing anymore.
Oh. Now you can see?
Yeah.
Yes.
Okay. So we type TF3A. Then here we type the name of the organism that we are interested in. So I'll do Oriza.
For those of you who need the link of NCBI, it is on the chat box.
So I'll do Oriza here. So nothing is found. So what we need to do is we need to go for model organism. Then Arabidopsis is a model organism. Then I type Arabidopsis here and do the search. Okay, now I can find the genes TF3A in Arabidopsis. Okay, I just click the first one. And here is the gene ID that I'll copy that one. And one thing we can do is like we can just blast in NCBI by opening a NCBI blast. Or we can go to phytosome, copy the sequence and go to phytosome to extract the sequence. So to nucleotide blast, just paste the sequence here and choose the organism. It is rice here, so I'll just do the blast.
So there are two questions, some comments here, Dr. Parajuli. One is, can you please zoom a little bit so that they can clearly see? The other one is, if you can slow down a little bit, as you know, they can follow. So I'm not sure how much time we have. So based on how much you have to cover, maybe you can slow down a little bit.
Okay, I'll just repeat. Okay. I can just stop. So go to NCBI and type TF3A first.
Can you zoom in on your screen?
Is it fine?
Yep.
Okay.
Perfect.
So I'll just repeat it from the beginning. Okay, I'll just type TF3A. So there are maybe that is for error purposes. I'll go from the beginning. TF3A, and I need for rice. So if I need for a specific organism, then I go to advanced, then type TF3A here, TF3A, and horizon. So I know like this gene cannot be found like this, so there is another technique to find the gene. So I'll just go for model organism, Arabidopsis, and do the search. So from here, I'll just get the first gene that I can see on the top. And if we need to blast that gene for rice in NCBI, then we can just copy this gene ID and blast it here. I didn't see anything. Okay. So we'll get the sequence of this gene. So we scroll down, and here is different kind of format: gene bank format, FASTA format, and sequence viewer. I'll just get the FASTA sequence. Copy the sequence, and if we want to see the gene bank file, then we can just click here.
In the next talk, Dr. Paudel is going to talk about different file formats, where he will talk about this FASTA format or FASTQ and other formats. I'm not sure if he's going to talk about the gene bank format, but this is what gene bank format looks like. So maybe that's.
This one has a lot of information. All the information that you can find here, like exon sequence, intron sequence, you can see everything here. And you can see the protein sequence too. This is the protein sequence. So this is the NCBI gene bank format. So I have copied the FASTA sequence. So now I'm going to next website, Phytosome. Dr. Paudel, can you copy and paste the? Let me do it different. Right. So once we go to the Phytosome website, then we can look for different organisms. Like most of the plant genome are here, and we just look for the genome of our interest. So now we're looking for rice. So we go for Oryza sativa. So this is the genome of interest for us now, and we just paste the sequence right here. Sorry. Just copy this sequence and paste in Phytosome. And okay. Let's get the first one. Click here. then it will show you transcript sequence, and we can go and say this is the gene viewer, and we can go to see the transcript sequence. And now you can see the functional annotation that is right here, transcription factor 3A, and that is the functional annotation. And this is the genomic location in the chromosome, and we'll go for sequence. Now we can get the genomic sequence here. So this is the genomic sequence. It has UTR, exon, intron, and 3' UTR. And we can also copy the transcript sequence from here. And we can look for CDS sequence, and we can also see the peptide sequence. And if you want to download multiple sequence, then we can add the sequence of interest to the cart, and after adding we can download. So how much we can, like how many sequence we need, we can just go on adding the sequence, then finally we can download all the sequence at one time from here. Now I'm going back to the slide. So these sequences were from the phytosome. So you can see the genomic sequence, you can see transcript sequence, CDS sequence, and peptide sequence. These are the same sequence that I showed you in the phytosome website, and that is from the TF3A gene. And you can retrieve those sequence from these links. And in summary, for the basic of the molecular biology, so the DNA consists of 3 different components: sugar, phosphate, and the nucleotide base. There are 4 different types of base for DNA, but RNA consists of ribose sugar instead of deoxyribose sugar, and the nucleotide base is 3 bases are same, but it has uracil instead of thymine. And the protein has all the amino acid sequences which comes from the codon, and they are made with the tRNA, which has the anticodon. And a gene consists of promoter, 5' UTR, exon, intron, 3' UTR, and poly A tail. And so start codon is always the beginning of the first exon, while stop codon is the end of the last exon. So this is the end of my slide, and I have one bonus slide for you. That is the SnapGene viewer. You can use this software. This is free software, and by using this software, you can download the sequence and you can copy and paste those sequence in this program, and you can play with the sequence. You can see the complementary sequence. You can see RNA sequences. So let me share this one with you. Can you see the SnapGene viewer?
Yes.
So this is a plasmid, but I'll just talk about the sequence. So after you get the sequence from NCBI or phytosome, you can open this software and you can paste. After pasting, like if there are some annotation, it will ask you if you want to automatically annotate the sequences. Then if those annotations are available in this software, then it will automatically annotate. And if you see, some of them has been translated into protein. So these are the DNA sequence, and these are the protein sequences in the, let me zoom in. So DNA sequence is on the top, and these are the amino acid sequence. So the first one is methionine. It is ATG, and it has coded for methionine.
I think we are not seeing that. We are just seeing that circular plasmid.
Now?
Still same.
I don't know. Let me reshare it. Can you see now?
Yes. Yes.
Okay. So after getting the sequence, you can copy and paste and you can just play with the sequences. And if the annotation is available, it will automatically annotate. It will ask you, and you can let it annotate automatically. So this gene has been translated. So it has already identified the gene and it has translated into protein. So you can just click in the gene and you can see the details. So the total size is 630 base pair. It has shown on the top. I don't know if I can show it. And it says it has 63% of GC content. And the first codon is methionine, that is coded by ATG. And as we go towards the end, and there are multiple codon, and let's see the stop codon. So it is denoted by asterisk, and it is TGA. So TGA is the stop codon, and it is the end of the protein sequence. And I can see the details here. So there is the name of the gene on the top.
While Dr. Parajuli is showing that, can somebody write what are those different annotations you are seeing? So many different annotations you are seeing, different names. What may be those?
So this is the overall map, and this is the sequence. And we can also see the enzyme. Like if you want to do restriction digestion, or if you want to do some more experiment and look for the enzyme, or if you want to do cloning, then you can just copy the sequence, and you can also look for enzymes. And there are so many different things. You can design the primer, and you can add a feature. If you know the feature, you can add by yourself. You can just select the sequence and add the feature. You can edit the feature. So this is the free version. So what you cannot do here is you cannot insert a new sequence, and you cannot delete a sequence from there. But if you get the paid version, which one is very expensive, then you can do everything. You can copy the sequence, you can paste the sequence, you can take the sequence out. So many different things can be done. Even if someone is interested in cloning, it can do the cloning too. And based on this one, you can design different primer, and you can clone. You can make multiple plasmid, and you can even simulate the agarose gel electrophoresis, like how the sequence will look like in the agarose gel electrophoresis. You can do so many different things. So download this website. This is free to all, and just play with the software. And if you need any help, you can talk with any of us. We'll be ready to help you all. So this is the end of my presentation. Thank you so much.
Dr. Parajuli, while you are going on that one, can you please discuss a little bit about the orientation, sequence orientation, and also the negative positive strand and complement versus reverse complement?
Okay. Let me zoom in. Okay. So can you see it clearly? Is it clear enough?
Yes.
Okay. So these are DNA sequence, and if you see the top strand is starting from 5 prime, and this will be the 3 prime end. So this is the DNA sequence. And if you look the bottom strand, that is starting from 3 prime to 5 prime, then this is called complementary DNA strand. So it will be just like as we talk in the base pairing, A is pairing with T and G is pairing with C. So the complementary strand will be the strand where the base is pairing with another nucleotide. So if there is A, then the complementary strand will be T, and if there is G, then the complementary strand will be C. So likewise, we can move ahead and we can make complementary strand by ourselves. But if we talk about reverse complement, then it goes from the opposite direction. Then suppose we are designing a primer. So if we have to design a primer and amplify the fragment, then one primer has to be in the 5 prime end and another primer has to be in the 3 prime end. So the forward primer, for example, would be this one, the top one, AGTAC continuously 20 base pair. This will be the forward primer. And if we want to make the reverse primer, then there could be confusion. Like we cannot make the reverse primer from here, from the top strand, or we cannot make from this side of the bottom strand. So what we need to do is we need to make the reverse primer from the 5' of the bottom strand, because always the transcription starts from the 5' end. So this will be the 5' for the bottom strand. So if we move forward, so this will be the forward 5' to 3' here, and A, G, G, A, triple A, G, G, G. This is going to be the reverse primer, and this is called complementary DNA, reverse complement, because it is complement, but it is starting from the reverse side. And we can see so many different enzymes, and even we can choose the enzymes, and we can choose the enzymes based on the company also. So we can save our enzyme set, we can choose our own enzyme set, and we can see commercial enzyme set, and like cutter, 6 cutter or unique cutter only. So if I go for unique cutter, it will only show the enzyme that will cut at only one position, and it will remove all the enzyme that will cut at multiple locations. So if I click here, then most of the enzymes are gone, and we can see only few enzymes that will cut at a single position only. Yeah, I think that's all. Okay, so if you have any specific question with this software, then you can type in the chat box.
It's really interesting that you are showing this cutting edge, and I didn't know about this software. This even looks better than what Vector TI or something. That's the paid version I used to use. So.
Vector TI.
Yeah.
Vector TI is also a good software, but it is more interactive.
Yeah, I really like this one. And looks like you are using with the Cas9. So seems like there is some IC's Cas9. So great to see the genome.
This is the plasmid for genome editing.
Yep, great. Okay, so if you have any questions, please write in the chat box. So now we'll move to the next one, and Dr. Paudel is going to discuss about this file format. So basically, how that all these concepts in the molecular biology are actually seen. We see them as a text format or different format.
Perfect, thank you. Can you see my screen?
Yes.
Okay, perfect, thank you. All right, hello everyone. So now I will talk about the different types of files that we generally use in bioinformatics. So as Dr. Parajuli already showed you, he showed you some sequences, how to copy, and now we are going to see when you save those sequences, how should you actually save them on your computer. So the first and the most commonly used format is what we call a FASTA format. So a FASTA format is a simple way of representing nucleotide or amino acid sequences and proteins. So basically, if you take a look at this cartoon or picture right here, you can see that it has a lot of lines considering ATC, ATGC. You can see all of these. So these will be your individual nucleotides. And if you take a look closely at the starting point of this file, it has a greater than symbol. So all FASTA files, they start with this symbol. And after the symbol, this is called a header, and it can be, usually you put a lot of sequencing details, for example, where this sequence came from. Like for example, in this scenario, we are using human chromosome 13 sequence. And this is customizable, so you can, if you have a gene name or if you want to annotate it with something else, you can do it. Basically, it has to always start with a greater than symbol and then the name of this sequence. And on the bottom of that will be your actual sequence. So whenever you are sending your samples after DNA sequencing, and then you receive your data from a sequencing machine, I mean, you can get your FASTA. I will talk about a FASTQ file after this, which is the file that you first get. And you can see this type of FASTA file in a lot of sequences like a reference genome sequence or sequences of your genes, amplicons, primers, or your sequencing data that comes from Sanger, Nanopore, PacBio, or any other sequencing technology that you use. And all the FASTA files, when you are saving, like for example, if it is a Word file, you save it as a .docx, and for FASTA file, we usually save it as .fasta or also .fa. And the next commonly used file format is called a FASTQ format. So in the previous one, it was a FASTA, and now it's only a FASTQ. So the addition of Q on this means that this file has quality information as well. So in general, a FASTQ format, there are 4 major lines, 1, 2, 3, and 4. So this would be for one individual sequence read. Let me annotate this. So again, the top line here, it starts with @, and then there is other sequence information related to this. So this will be our header. But the second line that is right here, this is the sequence that, this is the fastest sequence that we can get. Okay. So on the third line is a plus symbol. And on the fourth line, you can see that there is just a lot of characters. And in fact, these characters actually code the quality for each of the individual nucleotide sequence that is up there. And this type of raw sequencing data, it can come from Illumina or 454 or any other sequencing technology. So if you take a look at it more closely, each individual nucleotide here, it has a corresponding quality value right here. Okay, these are coded in the computer, and the computer knows like how good the quality is for each individual nucleotide. And like when we are doing analysis, what we do is we first have this FASTQ file, and then we take a look at the quality of each individual nucleotide base. And then if we see that the base is of really low quality, then we trim those sequences off. And finally, we get a FASTA file, which I showed you previously, of a FASTA file that consists only of the high quality individual sequences. We move ahead with aligning and mapping those sequence reads to our reference genome, and the output of that file type will be called a SAM file. It is a short form for sequence alignment map, and it has a binary sibling called a BAM file. So it has the same information, but it is in a binary format. So a computer can read it, but it is not human readable. It is just done so that you use less storage while using those files. So in general, a SAM file will have a header section, which is on the top right here. It gives you the information on the type of chromosomes that you have in there. And the actual data is right here. So this would be data for one read that we have aligned to our reference genome. And it has a lot of information. For example, it has the position right here, and it has the mapping quality, and it also has sometimes it also has the actual sequence that was used during when you were mapping with the reference genome. And with it has a lot of other characters right here. These are other features that we use like while filtering to get like more high quality. Then these are coded with these systems, and when we are running different analysis, we use these different parameters in order to filter our data to get the type of data that we actually want. Once we align, then we move ahead with calling SNPs or indels or other things. So, and the typical way to store those type of format is in a file called .vcf, and VCF stands for a variant call format. So this is again just a regular text file that is regularly used to store gene sequence variation. again, you can see the top portion right here. These are all headers, and these have a lot of background information about what is done here. And specifically, there are different type of codes that are used, and the description for these codes. And these codes are actually used in here to show you, what a DP means, depth, and those kind of things. So the details are up here. And the bottom, like out of the header, the bottom part, this is what we actually have for a variant call format. It has the number of the chromosome, the position, the ID of that SNP or at that locus. We have information on what type of nucleotide is present in the reference genome and what type of nucleotide is present at the alternate allele. We know the quality of that SNP or quality of that locus, and then there is a lot of other mapping quality parameters that are right here. And in the end right here, this you can see N1, 2, and N3. So these are your individual genotypes. So if you are sequencing, let's say, different plants, then your samples would be right here. And the information that is given here, it is very tiny, but this is the only information that you will need. What this information shows is, it shows that at this locus, it will show what type of sequence, whether a reference sequence is there or alternate allele is there. And it will also show you the depth and other features that are described up here. So this is a very intense looking file, and it has a lot of information that we really want. And we like this file specifically because it has all the SNP or variant information, and then it doesn't have any sequence information. That means this file is very small, and it will use less computational power. The other common file format is called a .gff file. So this is also known as a general feature format or generic feature format. We use this file when we are trying to describe genes and other features, like for example, of DNA, RNA, or other protein sequences. And I have put the names of each of the column names that are here. So for example, a GFF file will always have the sequence name here in this location, the first column. It will give you the source. And then the main interesting feature that you need to look at is the feature file. We know, like, the sequence, if it is a gene or mRNA or exon, or if it is a UTR region. So this feature can be extracted later. And the other information that it shows, it shows the start point of that gene. So for example, in this case, the gene will start from 14905 on the sequence contig 106. So, and it also shows and where it ends as well. So from this too, you can get the length of the gene. If you are looking into mRNA, then you can go and just filter the mRNA right here. Or if you are interested only on exons, then you can filter those exons from right here. And in the end, this is attribute. So this is kind of a comment that you can put. And in general, we try to put the name of the gene that we have here. So for example, in this case, the name is given is, this is from cowpea, a cowpea GFF file. So this is Vigna ungulata. So this is just a short form for given to that one of the name of the genes. And the other commonly used file format is a .bet file. So bet format, the full form is called browser extensible data. It is again a text-based data, and it also stores. It is also used to store genomic regions. But the only thing is that it has a chromosome and a chromosome start and chromosome end. So these three lines, the three columns are the most common or the minimum requirement of a bet file. But in addition to that, like, it can have other other information like strand and where the while visualizing where the thick start of that gene sequence you want to show, and it shows those type of information. Again, this holds, it only has 3 columns, so it is a very compact type of structure that we commonly use. with that, this is the last slide, and these are the most commonly used file format that we use in bioinformatics. There are, of course, a lot of other file formats that we haven't talked about today. I mean, you can go to the UCSC Genome Browser in order to learn more about those other types of file formats. And people are, as we have new DNA sequencing technologies coming up, we have new file types coming up as well. So this is kind of a constantly moving target. So we have a new file formats coming on, but these are kind of the most basic and general file formats that you will commonly encounter while doing bioinformatics. That is my last slide. So if you have any questions.
Thank you, Dr. Paudel. So we see some questions, for example, from T.N. Busal. Is there any threshold to determine the quality of sequence in the FASTQ file? So I can answer that too. So generally in the Illumina, so it depends on the which sequencing technology you are using. In Illumina, in HiSeq, it generally has 0 to 40, and these are represented by different letters or symbols in that FASTQ file. And generally, anything higher than 30 is considered lower than 30 score, higher than 30 score is considered is good. So, and we can use a lot of different software, for example, FastP, FastXClipper, to get rid of sequence, something that doesn't have a good quality. And tomorrow, we are actually going to download one COVID-19 sequence and going to show you the quality of data, and we are going to discuss how the quality decreases when it is towards the end of the sequence and those kind of things. The other question is from Prasant. What does score signify in the GFF file and frame? I think it's, yep, go ahead, Paul.
So the score is just a floating point value that is assigned to each gene. And what is the other question?
Frame.
The frame would be the base feature. That would be the where the codon started, whether it started at 0, 1, or 2. So when you are translating that, you can have 3 locations on your FASTA file where you can start translation. And so if it starts at the first, it will be 0. If it starts on the second base, it will be 1. And if it starts at the third base, it will be 2.
And there is another question from Satish. Is HapMap a file format?
Yes, HapMap is also a file format, and when you are commonly doing a lot of bioinformatics stuff, these days HapMap is getting popular, and it can be easily converted to a VCF file, and VCF file can be converted to a HapMap file. So they are interconvertible, and a lot of output from Plink, which is also another software that we use in population genetics, and these are very interchangeable, and depending on the type of software that you are using, you can use either of them.
Okay, and I think one question again about the float value. When can this floating value be used? And I think more, I can answer that. I think this mostly used for the visualization purposes. I think that's all the questions we have. So we'll move on, and we'll have some break here before we go and another half an hour or so we'll talk about the basics of the Linux. So before that we'll take a break, and I would like to invite Dr. Himal Luitel to introduce Dr. Navraj Devkota, and we'd like to hear some perspective from Dr. Devkota on AFU research.
Thank you very much, Ananta. Now, before next session, I would like to introduce Professor Dr. Navraj Devkota, who is the Director of Directorate of Research and Extension at AFU. I would like to request him to mention how AFU can utilize the experts in diaspora and how we can jointly move in this regard. Professor Devkota, now floor is yours.
Thank you, Dr. Himal. Good morning, everybody. Namaste also. I've been enjoying the part of today's presentation also, but I think it's because of several other reasons I could not continue since the last 3 days. First of all, I like to congratulate all the organizers, host, co-host, and the NAPA families for this very wonderful sort of series of webinar, where lots of students, faculty, and other researchers are getting benefited from this novel approach. Thank you, everybody who has been involving in this process since the last few months ago. Right. Briefly, I just like to share a little bit about Agriculture Forestry University, the diaspora and the environment where we all scientists working in the areas of agriculture, including livestock, veterinary science, forestry, natural resource management. I think it's a quite good opportunity to be involved with Agriculture and Forestry University because of several reasons, but I like to highlight two main reasons. The first is it's the only one government-funded university, a technical university in the country with a long history of battle and then struggle. Our ancestors, our seniors, we all people fought a lot in line with establishing this university, and that we wish that the dedicated Agriculture Forestry University would do something for the development of science and technology and the generation of required human resources in the country. So in that sense, this is a pride to all of us. In that sense, also it's a common forum, not only for Nepalese or other scientists scattered all over the country and the globe, but particularly those alumni people and then the students or the faculty who have been working and all are affiliated in different parts of the environment. This is a matter of pride that we can contribute work from different perspective. Like, we can be involved directly in a collaborative sort of research. As you know that we are very open in terms of establishing research activities for a joint bidding process, for particular types of problem solving research in the area of agriculture or any sort of problem solving research. We have a good pool of faculty members and then also the students. The total amount of number of students nearly equals to 1,400 at present, including the undergraduate students. And also we have expanded out the environment like Agriculture Science Center. It's a kind of outreach program for generating and disseminating technology to the different parts of the country in collaboration with the Science Center people, the government people, the NARC people. So that's why outreach program, we have already established one center in Makwanpur, Palung and the Galchok in Gorkha. In the near future, we are expanding this program to Satyavati district, Dading district, and also in Padam Ghat of the Kalikot district. So we have opportunity of working through this science center. There also the research scope with our faculty members and also our senior students involving in master's or PhD or undergraduate level. And besides that, we have constituent colleges like, inaugural speech, our acting vice chancellor, Sarda Thapalia, she had talked about all these aspects of possible expansion and also the execution of opening of the new faculty, kind of constituent colleges, starting from Rolpa to Pakhribas. So that is a kind of distribution where you can involve, we can participate according to our interest or choices. And that is also a forum for not only for the teaching, but also for the research and other areas. Besides, we have a kind of a good Nobel centers established. For example, we've been working hard to establish this biotechnology center for last 3-4 years with a very bright leadership of our young faculty, Dr. Kodaria, Dr. Himal, and going to be Dr. Saroj Sapkoda and other faculty members also. So this biotechnology center will be one of the excellent center for us. We are also trying to establish similar types of other precision laboratories covering agriculture field in the main area of Faculty of Agriculture and also in Faculty of Forestry and Faculty of Animal Science, Veterinary Science and Fisheries. So once we establish laboratories and once we expand our program, once we have a good number of students and also the collaborating environment with those well-wishers, with those alumni people, with those scientists and researchers scattered all over the globe, we can do something novel, something contributing in line with advancing the agriculture research and development, focusing to Agriculture and Forestry University. I think I am not talking, as you know that I did not talk something new. Perhaps you might have some kind of expectation that I would talk quite different sort of things, but these are the things, that's the ground truth reality. If we work in this line, probably we can support and develop something to the building this of our nation. In that sense, these are the primary and simple things, but still we have not achieved, the expected goals and outputs because of the several other problems. In that sense, if we united, if we try to find our space, a kind of LOSI, working environment in association with Agriculture Forest University. That's a very good moment to all of us. And on behalf of Directorate of Research and Extension, and also on behalf of Agriculture Forest University, personally, I would like to thank you and welcome you, all the friends, colleagues, seniors, my juniors, my students, everybody, respected people who have been involving in these processes. I'd like to welcome you again, and then would like to expand hands of support, collaboration, and linkage in the days to come. Thank you very much.
Thank you very much, Professor Devkota. Now, I would like to call upon Dr. Ananta Acharya to continue the session.
Thank you, Dr. Devkota, and thank you, Dr. Luntel. It's really encouraging to hear from Dr. Devkota, and yesterday from Dr. Sada Thapalia, from all the faculties, and we are really, this collaboration we are trying to make, and hopefully we can do something. So thank you very much for your encouragement. Okay, now we'll go back to our technical session.
Thank you, Namaskar, for your encouragement.
We'll go back to our technical session. So for next half an hour or so, we'll discuss about the basics of Linux, and I'll invite Mr. Sishir Subedi to discuss about the basics of the Linux.
Hello, everyone. I think you need to allow me to share my screen.
You should be able to share.
Okay. Is it up?
Yes.
Okay, hello everyone. Thank you all for being here today. And for this session, we are basically going to start from very basic, like 101 of Linux. And we're just right away get into commands and discuss why these basic commands are used. And this will kind of give an overall warm-up for tomorrow's workshop session analyzing coronavirus sequence. So first command we want to discuss is ls, which is the lowercase l and s. So this command will basically show different file structure in the system. So you have ls, so it needs to be lowercase. If you have uppercase, then this will not work. So ls, and so right now I'm in guest in the system, and I have desktop just like Windows and other systems. In this terminal, you can see I have other download music, pictures, and templates and tutorials. So with ls, I can expand this command with parameter of a, which is meaning so everything. Sometimes there are other configuration files that are saved for user specific requirements, and these are bash, rc, and other ones. So if you want to see more, it's -a, or you want to see in a list format where you can get more information, such as the permission information, user, and groups, and when this file was created. So in a list format, then you have to type ls-l. And so if you have, sometimes like as you see here, if you type lsl, then you are given prompt at the end. So you want to have another command with r, so that you have the latest one in the table format with tutorial over here. So these are this one is the oldest one and this one is the newest one. So let's see, make some files over here. So ls command, just to look at different files. So we want to first start with making our directory, right?
Mr. Subedi?
Yeah.
Yep, can you please wait? This one you can also follow through while Mr. Subedi is showing this one.
Okay.
And I know that probably more you have not installed mobile XTOM and you have not accessed our SSLs. We have not shown how to do that yet, but I have shared a link there. It's https repl.it languages at bash. It is just the interpreter. It looks like what Mr. Subedi is showing here. So you can actually type all those commands over there and kind of see what is happening. So you can actually follow him over there. Thank you.
Yeah, thank you. So let's start by creating a directory. And to create directory, command is mk make directory dir. So, and then directory name, right? So right now my directory, I want to make, let's say, Corona directory. So now I want to check if that directory is made or not. ls Corona is there. So if I use ls ltr, then Corona is just here. So I just made. So now I want to go inside this directory. To go to this directory, I have to use this command called cd, which is change directory to Corona. So in Linux, it's very useful key here is tab. So I know I want to go to Corona. So I just type C and then press tab. Then it will try to find all the possible combinations. And if there is only one that starts from C, then it will just directly full itself, right? So if I want to do D, then there are 3 options. Then I can do narrow down by D, 2 option, and then O, now it narrows down, and then W, tab. So I'm pressing tab, right? So let's go back to C, Corona, change directory to Corona. I mean ls, there's nothing here. So let's make another directory, mkdir Corona. Let's call it D1 for depth 1 inside Corona, right? So I'll make this directory again, ls Corona directory is here. And I want to again go to inside this Corona D1 directory that I just made. Corona tab, there's nothing here. I can use ll also, which is more I'm used to, but ls is more basics. So now I want to make another directory inside Corona D1. So Corona D2. So this will be depth 2. And ls here. So I have now Corona D2, change directory to Corona D2, right? So with this one, now I've been making this many directories. So I want to know what the path of current directory, and command for that, print working directory, pwd. So if I type this, so now I'm in, it shows here from home. I'm right now logged in as guest. I made Corona directory, and then depth 1 Corona another directory, and then Corona depth 2 directory inside D1. So it will show me all the path where I am. So with this, as we know, we can use change directory, right? So there is a use of dot here. So if I can use 1 dot, that means I am at wherever I am, my directory, I'm not going to change. So cd dot here. And if I do this dot twice, 1, 1, it will get me back to my parent directory, D1, right? So now I can do pwd print working directory to check. Now I am in D1. So now again, cd, I want to go to back my parent, then pwd, I'm in Corona, right? So let's go more over here. So now we can go to our all the directories like C, Corona, and then since we have only one directory, we can keep pressing tab, Corona dep1, Corona dep2, and that's it. We are in PWD in our directory. So if we want to just go back to our main home for guest user, then we can just type cd, and it will just get back to our main home ls, where we have first Corona directory. So this gives us how to see different directories and files, ls, and some options here. Dash a will give you all the files. Dash ltr in reverse format. Corona. Cd will make new directory, and then cd will go to change directory. Mkdir will make new directory, and then finally pwd will show our print our working directory. Now the next is we want to talk about copying files, right? So now I have a few files here, which I downloaded for tomorrow's session. So download. So there are COVID samples, and this is zip file. I unzipped it, and there is COVID sample. I want to go and see what's in there. ls. I have GISCD files. There's some scripts, right? And I want to now take this COVID sample here. Let's look at this sample here. So here, COVID sample FASTA file, and I want to transfer this to Corona directory that I just made, right? So in this case, I can now go back to I know I want to take this file over there, and simple is first you go back to your home by typing cd, then we go to Corona inside ls. This is another directory, but we want to bring this file from downloads to here, and command is cp. And now what we can do is this is a path. We know we have to go to home, and then I am guest right now, and then I have that in my downloads, and it's in COVID samples. And so I'm pressing tab, so that will just show me different options what's in there. So I'm typing COVID FASTA file here. So now I want to copy that to Corona, and as I mentioned earlier, to show that where I want to copy, I can copy as is, or I can give my file name. Let's just file name it as COVID FASTA over here. Yeah, so now we can check it's COVID FASTA. It's here. So now we have copied our file from another directory to this directory, and we use cp command, right? Cp command, your source where I copied from this download location to destination, which is my Corona directory. Now, next thing I want to do is I want to, let's say I don't want this file. I want to make first another copy of this here. So I can just do copy COVID FASTA here, and I can just make a copy, meaning I can just make COVID version 2.FASTA, and I just copied my files, both of them here, right? So copy will work because this is also source. I have this original file here, and I just want to make another copy, and my destination is also same directory, right? So this should work. And now, I don't want this file COVID v2 that I just made, and I want to delete it. And for deleting is rm remove file name, so COVID and v2. I don't want this, so it's just rm remove command and COVID version 2 FASTA. It removes it and make sure it's removed. So I'm back to my directory, Corona main directory. I have one file, COVID FASTA. Now I want to look at this file. So it has many sequences from 20 different samples that we'll be using tomorrow. So one command is cat is just view file, entire file at once. So if I do COVID FASTA, then it just, everything came up, right? We didn't get a chance to look at it. So this is because of this. If there are small files, only a couple of lines, then this will work. But then if you want to look at like we're looking book or copy page by page, then the command is less, then it will show page by page. And then I can just enter through and look these different sequences.
Just want to add one more thing. Less sometimes may not be available in your version of Linux. I think in the example I have in that website, if you try less, it may not work. In that case, you can try more. More might work.
Yeah, so more is just another. So what percentage of we are viewing it? And like there are many information in your terminal. So I want to clear this up and start from the beginning. It's clear. It's literally clear. So start here. So now I want to, so list gives us page by page, but I just want to look first few lines that is head of that file. So that is basically head, right? So it will give me this many lines, and I can be more specific by saying, okay, just give me 5 lines, and that is -n5, number of lines for -n, and then 5. So 5 lines. And similarly with tail, I just want to look at the end of this file. So it will just give me this, and if I can. So another thing is I'm going back by up arrow. So instead of typing, if I want to go back what I typed earlier, I can keep going back. Right, so I can, now I don't want to type clear, for example, then I can just keep going back. Oh, I typed it earlier. Then I can just get this clear code command here by just pressing up arrow key multiple times. So clear this. Right, so I did tail earlier. Now I want to -9, and let's say I just want to do 4 lines here from tail of this file. So now we have, let's go and look this file again, COVID FASTA. So we know, we see this is a file format, FASTA format that we already discussed during presentation. And this arrow key here, greater sign, gives reference or whatever the name of this sequence is here. And currently this file consists of 20 COVID sample sequences from different countries. Now, what I want to do is I just want to look at these names. I don't want to look at the sequences because it's hard to look in this format. We want to visualize and use other tools. But I know that all these sequence names are given, the first character is this sign, right? Greater than sign. So in this case, there is another command called grep. So it is used to find some portion of file that we are interested in. And now we have to say what we want to find is this sign. Wherever this sign is there, it will print that line, right? COVID FASTA. Okay. Now we can see that there are so many, it's 20 sequences from Nepal, Wuhan is referenced, India, Pakistan, Italy, 4 from USA, Sweden, and this. So this will be what we will be visualizing and analyzing tomorrow, see how they are related and all that. So for now, like we got rid of all the sequences in middle. We're just interested to get these names. So that for that command is grep. So now I have this information, right? So I want to keep this information separately. I don't want to keep doing less or look into this file every time. So I know I can get this information, but now how do I put it in somewhere else? And that is called just redirection, and redirection is just one arrow here, and I can give a file name with this portion what I just displayed in standard output here. Then I can redirect you to file name, let's say COVID sample names, sample names.txt. Okay, so now if I go and open that, now I can do cat because I know this file is not right. So this is from COVID. Yes, and I can also do less to look at this. And since this file has only a few lines, we can see everything here. So this is one, but now there is this, we can redirect this. This will just make new file every time when we use only one arrow. But if you want to append it to another file, let's say if we want to append the same information again, and then the way to append it, just add another arrow here. So now it will make it twice, right? Because we appended. This was the from first part with one arrow, and then we use another arrow to append it. So that we used was this two redirection symbol. So another command is we want to count how many lines are there in our file, and that is command is word count and our file name. So COVID sample. So we have 42, right? So it just shows 42 lines, and then we can see these are words and character. So 42 lines, 84 words, and 1564 character. Or we just want to get a line, then there'll be 42 lines in this file. Okay, Dr. Acharya, you have some any other topics?
No, I don't. I think you covered pretty well.
Yeah, so this is just a basic, just to get it started with Linux, and just the basic commands like just revise ls, copy, mkdir, right? And then remove, word count, grep to find out how many to specific lines out of file. So these are very basic, let's say 10 commands or so in basic Linux.
And that's great. And I think this will get away. We can probably go with just these commands and some other commands, especially for those specific programs. For example, aligning and those kind of things. We'll introduce new commands, but for basically Unix operations, these commands should be enough for now. And if we see something new tomorrow, we'll always can come back, and then discuss about those. Okay. So we have 10-15 more minutes, and now do you want to cover more anything more on the Unix, Mr. Subedi?
I think
yeah, if we were done, I think we can kind of take any questions, and also we can also do two things we want to show. One is how to download that, and also we want to show you how to download the MobiXTOM. So because that will be used for tomorrow, so we'll show you how to do that.
Sure.
Okay. Dr. Parajuli? Do you want to share your screen and show where they can? Yep.
So I think there was a lot of confusion between the Snapgene and Snapgene viewer. So if you go to Snapgene, then it is not free software because it has a lot of feature. And I'll just open both.
Can you zoom your screen a little bit?
So this one is Snapgene. So if you go here, it will say free trial, try for free, and pricing. And if you go to Snapgene viewer also, on the top it will say Snapgene free trial. So a lot of you are confused about like if you have to download free trial or if you have to pay for it. But this is for Snapgene. But if you see here, this is Snapgene viewer. This one is free. And it has written here because there should be no barrier to seeing your data. So this is actually free. Don't go here for free trial, just go down. And if you are using Windows, use this one, Mac, Ubuntu, you can choose your operating system and you can download it. So if you click on, suppose Windows, then it will automatically download the file and you can install it. It should be free. So don't be confused with this one. So this one is Snapgene, but what you have to look for is you have to look for Snapgene viewer. That one is free. Yeah. Any questions about this one? Let me know if you cannot download it.
Okay. Thanks, Dr. Parajuli. Now we are going to show you how to install MobaXterm. Dr. Paudel.
Right, can you see my screen?
Yeah.
So just search for MobaXterm. Okay, and it says MobaXterm free X server and tabbed SSH client for Windows. I am on a Windows machine, so it will say it will be Windows. So if you go and see this, you can go down where it says get MobaXterm now. Go ahead and install the home edition free. This is the free version. You can download this. And there are two types of MobaXterm. One is the portable edition, which you don't need to install, so it will work automatically. And the other one is an installer edition. You can do either of them. For mine, I'm just using the installer version. And I can unzip it to install it, but let me download the portable version as well. Looks like it is getting stuck.
Yeah, I think everybody can do that way. So
yeah.
Maybe you can just open the MobaXterm now and just show some interface and how do we connect to the cloud.
Okay, so when you once you install MobaXterm, so this will be something like this. Let's see if I can change the settings on the display. Okay, I think it is a little bit bigger than before. So this is how you will see your the interface for MobaXterm. So that we'll use tomorrow. And if you go to session and new session, so there is a lot of different things that you can do here. And tomorrow what we'll be using is something called SSH. So tomorrow we'll give you the remote host name, the username that you are going to use in order to log into the system that we have prepared in Google Cloud. So this will all be available to you when we start the seminar session tomorrow. So once you are in that system, we all will be using basically the same system. So all the files that you need will be there. We'll provide you with the different types of commands that we will be using. So you will be required to create your own directory, as Sishir showed you earlier today. And once you have your own directory, we'll have you copy the data that you have already downloaded into your directory, and then we will start the analysis part after that. So this is where we will start. So I encourage all of you to download Mobile Extreme. And there are other tools like Putty, and if you are on a Mac, you can just use Terminal to access this. So I think that is all from my side. There, I can see a few questions in the chat box. Okay, first question is Putty and Mobile Extreme the same? So Putty and Mobile Extreme, they are two different software. They have different uses, but most commonly, if you are only using them as an interface, they are exactly the same. You know, you can do the same things in Mobile Extreme that you do in Putty. The only advantage of Mobile Extreme is you don't need another interface when you are transferring a file from your local computer to the cloud. And also you can view everything like directly in the Mobile Extreme as compared to Putty. So, but if you download Putty, Putty is a much smaller file size. So if you have like problems with internet, you can still download Putty. But basically, once we log into the system, it will be the same if you use Putty or Mobile Extreme. And another question is from Rishi Khatwi. It is, can you put the detailed process of downloading Mobile Extreme? So I can go again to go to the Mobile Extreme website. So just Google Mobile Extreme, go to its website, go to where it says Get Mobile Extreme Now, and go to the Home Edition, which is free, and go to Download Now. Once you have that, you have two different types. I'm installing the version, the installer edition. You can go ahead and install the portable version as well. I think my portable version is already downloaded. I don't see it here. But yeah, you can use either of them. Both will look exactly similar. Okay. Yep. Dr. Acharya, if you want to share the same comment, I think it is very important.
No, I think you covered everything. So.
Okay. Yeah. And also, I know like you all are already overwhelmed with a lot of new things today, but we just want you to orient towards what you will be seeing tomorrow. So, you know, when you see everything tomorrow, it will not be, oh my God, what is this? So you at least know that, okay, these guys are tomorrow, they are going to come here, they're going to do LS, they are going to do PWD, they are going to do CP, and those type of simple things. So we will repeat all of this tomorrow again. So on our own system, so it'll be easier for all of you to follow around.
And I think I want to add one more thing. I still see like 149 people here. And what we are not really sure is if everybody wants to try your hands-on. We planned hands-on with smaller number, then we thought we'll do a demo. But again, we are thinking, we are really encouraged and seeing your participation. So we really want it to be the hands-on. That is why we shared some of those links and we are sharing this Mobile Extreme. So if you really want to, so we have to send you some key file so that you can connect to the our server. We cannot send it publicly, so we have to send one file. So to send that file, we need to know your email address. So I think Dr. Paudel, how do we want to do that? Do we want to make up a Google form, or do you want to give one of our email address so that they can send their email address and we can send them the private key file?
I think it'll be easier. Do you want to send to all of them? What do you want to do? Because if we send to all of them, probably it'll be the most easiest way. So we don't have to.
Okay, then let's do anybody. Okay, then let's do that way. Okay, so
yeah, I'll send an email to everybody to for about the login details for tomorrow.
So we can do in the chat box tomorrow, right?
Yeah, don't worry about sending your emails. I should have all of your emails when you registered for the seminar. So if you have received an email from me saying that the seminar will happen at this time, and you have received a Zoom link from me, you will receive an email again about the login details.
So again, so just want to add on that, I know some of you have not registered, but you are attending today. So if you are one of them, please, and I think from this chat box, we may not be able to track all this. So please send your email to my email address. That will be ananta. I'll send that right here. scharya@gmail.com. So if you have not registered in the registration, send it to here in my email address so that I can send to everybody.
Yes, perfect. Do you have any other questions? I think we are a little bit early and on time today.
Yeah, this is great to be on time. Doctor, maybe we want to wrap up with some comments from Dr. Kadaria, Dr. Luitel.
Yeah, Dr. Ananta, thank you. It was really nice today's presentation and hands-on workshop. I'm very happy you all are you are tirelessly you are trying to deliver some knowledge to our beginners and practitioners from EFU, from GU, KU, and other foreign university from abroad. So it's really interesting, really fruitful workshop. We are more than enough. It's our more than expectation. It's very fruitful training. So our higher authority, vice chancellor, our research director, and deans, they are also very happy. And so in coming days, so we are thinking more about coming days. When we are worried about coming days, how can we take your knowledge, expertise, and then your skills to the country? It's our concern, and that is really interesting. So I hope some beginners are feeling some difficulties to understand these big things, big content. So think about that. So how to involve beginners properly is my concern. Maybe tomorrow you will cover. You will give some way to go to beginners. Thank you, all of you.
I think we should announce one more time. Please email to Ananta, not in the chat box, because in email here in chat box may not be entertained to get the files. So you have seen this email of Ananta. So just email him to get the information. Because there are a lot of emails coming in the chat box.
Okay, we can take some feedback from our audience, our participants, if we have time. If time permits, 5 minutes we have time. If anyone can put some feedback or some suggestions to organizer?
Yeah, I think we have comments of thanking all the team for very informative session. And there was a question on the chat box about showing how the MOBA Xterm looks. So this is how it should look if you can see my screen. It will be something like this. So tomorrow we will start a new session, going to session, and we'll let you know what to do tomorrow. But so make sure you are
maybe they don't have access on that's muted so mute on, so they don't speak here. They don't share.
So let me add one thing, like if you have Mac or Apple computer, you don't need to download Movaxtrom, and you can download also. So you can directly go to terminal, you can open terminal, and you can do everything what you can do in Movaxtrom.
I think that will be it. We should move ahead with closing. Dr. Acharya.
Yep. Okay, so again, thank you very much. Thank you for your participation. We are really encouraged. Like we have said time and time again and again. We expected maybe 10 or 15 people doing the hands-on, but we are really encouraged seeing so many people on the hands-on and so many people on the sessions yesterday and the day before. A lot of the faculty members, a lot of the researchers, students, and everyone. So we are really encouraged. And let's finish this one with 2 more sessions, and then let's continue on working on together something really applicable from here and collaboration with Nepal. So that will be great. Thank you very much.
Thank you all.